We continue our survey of ion channels with the TRP channel. This is the so-called transient receptor potential channel, so named because of the phenotype that was seen in Drosophila, where the channel was first identified. This is an extremely large family of channels. There are over 100 genes that code for various types of TRP channels. The structure is similar to that of other voltage-gated channels. There are 6 membrane-spanning domains, and 4 of these subunits are needed to form a complete channel. However, there's no voltage sensing domain in this channel, so these channels are not voltage dependent. TRP channels are rather non-selective cation channels that are permeable to sodium, potassium, and calcium. Although they are not modulated by membrane potential, they can be modulated by G-protein coupled receptors, and this occurs through the activation of phospholipase C. TRP channels play an important role in sensory transduction, specifically in chemoreception. There are TRP channels that are sensitive and activated by capsaicin and methanol. For instance, the TRPV1 channel is sensitive to capsaicin. It's also activated by heat and pH. Other TRP channels are extremely sensitive to temperature. They have these specific types of TRP channels have a Q10 of greater than 10, which means that their open probability changes very dramatically with small changes in temperature. Other TRP channels are mechanosensitive and function as mechanoreceptors in sensory receptor cells that are sensitive to touch. TRP channels play an important role in development and specifically in the guidance of growth cones, and this occurs by a series of signaling mechanisms beginning with calcium influx in the growth cone, which can be evoked by various growth factors, and this in turn activates phospholipase C signaling. Another class of ion channels that play a very important role in sensory transduction are the cyclic nucleotide gated ion channels, the so-called CNG channels. These channels gate open. Their open probability increases when cyclic nucleotides bind to the C-terminus of the channel protein on the cytoplasmic side of the channel. Cyclic GMP is most effective in modulating these channels, although cyclic AMP can also modulate the open probability of the channels. The channel pore of CNG channels is largely non-selective to cations. These channels are permeable to potassium, sodium, and calcium. These channels play a very important role in sensory transduction in various sensory receptor cells, primarily in photoreceptors and in olfactory cells. And the effect of cyclic nucleotides on the open probability of the channels is shown in the graph on the lower right. When no cyclic nucleotides are present, the channel has an open probability of 0. When cyclic GMP is added to the intracellular face of the channel, the open probability is quite high. At 0 millivolts, it's about 0.8, and you can see from the relatively level slope of the graph that these channels are largely voltage independent. Another important channel which has somewhat anomalous behavior is the HCN channel. This is the so-called hyperpolarization activated cyclic nucleotide modulated channel, and it generates what is called the IH current. It is anomalous in its behavior because, like the inwardly rectifying channels, the open probability of the channel increases as neurons are hyperpolarized, and specifically these channels are activated at voltages below about -60 millivolts. The channel is permeable to both potassium and sodium, but unlike other types of channels, the permeability to potassium is quite a bit higher than to sodium. The permeability ratio for potassium to sodium is about 4 to 1. This means that the reversal potential for the channel is relatively negative, about minus 25 to minus 40 millivolts. Another interesting feature of the channel is that it activates and inactivates relatively slowly. Different types of HCN channels have activation or inactivation kinetics of about 50 milliseconds to a second or more. So these are very slowly activating and inactivating channels. The channels are also modulated and potentiated by cyclic nucleotides. And the channel has a very small unitary conductance of about 1 picosiemen. HCN channels have many important functions. As we'll see, they play a very essential role in pacemaker activity in generating oscillations in many types of neurons that have bursting behavior. HCN channels also play a role in regulating the resting membrane potential of neurons because they are open at the resting membrane potential, and they also can modulate the time constant and length constant of dendrites, thus modulating synaptic integration. The experiment shown on this slide illustrates some of the properties of HCN channels. In the upper left, we see a voltage clamp experiment where, in this case, the neuron is hyperpolarized to 103 millivolts for this trace, and we see that there is a slowly developing inward current. This is generated by ions flowing into the cell through the HCN channels. The current voltage plot in the bottom left illustrates the activation properties of the HCN channel. And as we can see, there's anomalous rectification where the open probability of the channel increases as the cell is hyperpolarized. This graph also illustrates the cyclic nucleotide sensitivity of the channel. When cyclic AMP levels are raised, the IV curve shifts to the right, indicating that the open probability of the channel has increased. So cyclic nucleotides, including cyclic AMP, can modulate and increase the open probability of the channel. The trace on the right illustrates a voltage recording from a sinoatrial node cell from the heart, and this shows that the oscillatory behavior of these cells can be generated by just two types of channels: the HCN channel, which generates the IH current, and a T-type calcium channel, which generates the IT current. If we look at the oscillation at this point in the cycle right here, the cell has hyperpolarized to about minus 60 millivolts. This will activate the HCN channel, and the cell will slowly depolarize as the HCN channel slowly activates. Now remember, the reversal potential for this channel is about minus 30 to minus 20 millivolts, so when the HCN channel opens, the cell will depolarize towards that reversal potential. When the cell reaches about minus 40 millivolts, this activates the T-type calcium channel. These channels open up, calcium flows into the cell, and this more quickly depolarizes the cell. The cell goes to a very positive voltage, and then over maybe 100 milliseconds or so, the T-type calcium channel inactivates, and then the membrane potential of the cell falls back to a hyperpolarized voltage driven by leakage potassium channels. When the voltage of the cell reaches minus 60 millivolts, the HCN channel is again activated and the cycle begins all over again.